Thank you. I'm S.Makeig, and I prepared this with my colleague, Bing Jung. We're at the Swartz Center for Computational Neuroscience at UCSD. And you may have heard of us through the EEG Lab software environment that for the last 20 years we've built and maintained, which is likely the most used environment for EEG signal processing by research in the world. So what are the advantages of functional brain imaging using scalp EEG recording? There are several. First of all, it has very fine time resolution. That is, we can record brain dynamics that occur at the speed of action and thought, quite unlike the metabolic brain imaging methods. EEG has increasingly good spatial resolution. Thinking otherwise is an outmoded idea, as I will show. EEG is completely non-invasive, and it's well tolerated by nearly all subjects, patients, participants. It can be lightweight, mobile, wearable, cheap, and portable, unlike, again, the metabolic and other brain imaging modalities. Therefore, it can be scalable for clinical applications outside of a laboratory or hospital setting. And while it was the first functional brain imaging modality, for nearly a century ago it was demonstrated. For many decades, the progress in its modeling and interpretation has largely stalled. Now, the origin of EEG, primarily in the cortex, is complex and not well studied. In the era of the single-cell neurophysiology, talking about field potentials was nearly forbidden. And so there's a lot of the complexity is not really well understood. We know that the connections between excitatory and inhibitory cell networks in the cortex, the cortical thalamic loops, the cortical connections, distant connections between sparsely connecting different cortical areas, and what Edelman called the value system modulations by the brainstem-centered systems using dopamine, serotonin, etc., as well as neuroglial coupling, which was a pet topic of my mentor, Galambos. And finally, the non-synaptic ephaptic modulations, which have again been shown to produce an effect not through synaptic transmission, but the effect of local field potential, extracellular field potentials on the timing of spikes. All these complexities regulate the appearance of EEG and then in its dynamics. As well as, of course, it has a layer structure in cortex. Now, the problem underlying the interpretation of EEG has always been and remains that synchronous activity within a single source area, simulated here in a forward model of a head, is said to resemble the projection of a tiny battery, an equivalent dipole, and its scalp projection looks like this in an actual head model. You see almost all the channels, except perhaps around the equator, midline, receive projection from each channel. So the point spread function for EEG is very broad. Or looking at it the other way, if you take a channel, and so we have a channel from electrode A to R here, and in a head model, then you want to say, what is the receptive field of that channel? That is to say, what areas of what cortical signals does it sum? On the right, we see the answer. The answer is that at time t, the voltage in that channel is the dot product between voltages in every portion of cortex, every mini column, if you will, and this masking function, in which only the bright green areas have no contribution to the channel. So when you're looking at activity in a scalp channel, you're seeing combinations of activity from all over the brain and not in any neat physiological network or system. This is the fact of life about EEG. Now, if you have Nowadays, MR images or even image sequences of images, and you can segment different tissue types that have different conductivities. Then you can build either a boundary element models or finite element models, electrical forward models saying how current should pass from the cortex to the scalp surface. And then given some EEG or MEG data and appropriate sensor co-localization with the head model and signal processing, if you find a suitable inverse problem that actually represents the simple map, that is the projection of a single source area to the cortex, to the scalp, then this problem is in most cases solvable to estimate what is the source area that was involved. However, finding this simple map is not so easy. So 25 years ago, I discovered work ongoing in the field of applied signal processing using independent component analysis, which separates signals into sources of information rather than trying to characterize variance in the multidimensional signals. And so we have single sources with different functional origins, summed in the scalp electrodes, and I see a decomposition will separate out, first of all, the non-brain sources of EEG signal. Here we have on the upper left vertical eye movements, on the upper right horizontal eye movements, on the left a particular muscle, postauricular muscle, and on the right the ECG contributions through a session of data. Once those and many others are separated out, what is left are sources whose scalp projections, learned also at the same time by the decomposition, resemble almost perfectly the projection of a single tiny oriented battery or equivalent dipole, equivalent to a patch of cortex, or sometimes two patches of cortex that are plausibly well linked anatomically or physiologically. Furthermore, the time courses of each of these processes is viewable with high resolution continuously. Now, can we find out any more about where these sources are except using a 3D dipole in an empty brain volume, template brain volume? Well, if you have an MR image of the subject, you can make a forward head model. The main, and we use SCS to localize the sources onto the cortical mantle. However, that's an algorithm that was developed by Cheng Chao in our lab. SCS stands for sparse, compact, and smooth. Now, the prime and most important missing parameter in all of EEG inverse source localization has been and remains skull conductivity. Skull conductivity in adults far outweighs brain conductivity of all brain tissues far outweigh the skull conductivity. And the skull conductivity in adults varies by, we believe, about 3 to 1. And if you go to children, infants, 3 to 1 in the other direction. So, and the effects of this on source localization are profound, up to 2 centimeters of depth information that is lost. So we've developed, with Zeynep, Kaylin Achar and I have developed the scale algorithm for simultaneous conductivity and localization estimate. What we do is we take about a dozen of these independent component processes. We treat these as projections of virtual electrode, intracranial electrodes. We simultaneously solve for their distributed source solutions. Here we did an experiment starting with two very different assumptions for brain conductivity. And then we iterate, building head models, following the gradient descent, and arriving here at a common solution in which the localization estimate is singular. And in this case, giving an estimate of the brain to skull conductivity ratio for this subject of 34. Now, using this SCS and scale technology, we can easily find and localize in cortex the sources of many recorded components. This was during a complex video game session, video game playing session, and looking at a site like Neurosynth, one can easily associate these regions of cortex with function, cognitive function, and in this case, all the labels that one finds immediately are in fact highly compatible with the cognition the subject had to exercise to play this game. You say, well, this is an hour of data, so these were the only places in cortex that had this synchronous local synchrony that produced a source. Well, no. If you use the non-stationary version of ICA, developed again in our lab by Jason Palmer, you can find many, many more of these sources that are active during periods in the game, and you can also see which periods they were active in. So now, about a dozen years ago, I thought to myself that it struck me that all the brain imaging modalities are require you to hold your head very still. And even in EEG, people were asking people to hold their head very still and stare at a fixation cross. Why? Well, in all the modalities except EEG, the sensors are very heavy, and any head movements will contribute intractable noise signals to the data. But of course, this is a very highly artificial limitation to brain imaging, because nearly all our life involves actions and interactions with 3D environment, and our brains have evolved to control our behavior and to optimize the results of our behavior. So it's quite plausible to say that you haven't really looked at brain dynamics supporting natural cognition until you study a much wider range of situations, perhaps not starting with modern dancers. So I thought this was an important enough concept to venture an acronym, MoBI, for Mobile Brain and Body Imaging, with the motto that you should record what the brain does via EEG and or other imaging, what the brain experiences versus via scene recordings, experiment recordings, and so on, and what the brain organizes, which is our behavior. That is, one wants to approach studying natural cognition in a much wider range of settings. And building on the concept ever emerging now in psychology of embodied agency, that all our cognition, including our abstract, even aesthetic cognition, is built on and grounded in our embodied experience of being an active agent in space-time and having agency within that. So for instance, in our lab, Hiroyuki Kambara from Tokyo Technical Institute came and did an experiment with me on juggling. And on the left, we see multivariate Granger-like causal functional connectivity modeling of activity in her brain as she was juggling. Now, what about development of new technologies to enable more broad range of experimental and recording situations? So here's the standard high density cap. This is a Biosemi 256 channel cap, which is optimized for use in the recording laboratory. The electrodes are plugged in, they're easy to wash, and so on. Well over a dozen years ago, I think people began building dry electrode sensors that did not require use of a conductive gel to make contact with the skin, so they used high impedance. And then first systems were made about a decade ago, low density and rather clunky. This was built into a headphone by colleagues of my associate Ziping. This is the 64 channel cap now offered by Cognionics, this UCSD spin-off company of Mike G, in which the sensors, in order to go through the hair, have little fingers. They're 3D printed little sensors or spiders that go into the head. But the system is wireless, it's easy to put on. And the technical challenge for building such systems has been and remains to achieve high reliability of the signal while keeping the user comfortable. You have to apply pressure to the electrodes, they have to go through the hair, so they have to have small fingers or tips. And yet if you ask the subject to wear it for a long time, it's very uncomfortable. So therefore you have to use some strategy to distribute the pressure over the whole scalp. And I've seen in the last decade this process ongoing. The electronics themselves are not so much a challenge, although, of course, continuing advances in cost and energy efficiency and robustness to environmental noise and so on will continue to go on. Now, Cognionics also makes a 1020 array. This is the array that was put in place for clinical EEG studies in 1940s, I believe, and remains as the standard now, held there by the insurance companies to some large extent, and by a tradition that the neurologist or clinical neurophysiologists are the keepers of the knowledge of what EEG means. By visual inspection, they can tell you. And if you ask what ground truth is, they said, I am ground truth. Well, I think this approach, although it may be lucrative for companies doing it, is not the approach that should be really sponsored. It should be, in fact, high density, where needed, wearable and wireless, energy efficient and comfortable EEG recording systems with optimal individual MR-based forward head models. It's possible to build low-cost imagers to make these images easy and cheap to collect, I believe. And they need to be optimized with conductivity estimates. One needs to record exactly where the electrode positions are. And nowadays, your cell phone will allow you to do that with the new 3D photogrammetry that's possible. Waving the cell phone around the head will record a 3D image of the scalp surface that can be co-registered with the head model. Then applies high-resolution cortical source decomposition and imaging, and model the non-stationary cortical dynamics and the concurrent body and eye movement behavioral dynamics in the so-called MoBI framework. I think together all these advances as they enter the field will really revolutionize the use of EEG technology and make it a true part of the portfolio of 3D brain imaging tools and modalities that will lead to new discoveries in human neuroscience, applications in medicine, as well as undoubtedly commercial applications. Thank you.